Alright, this is Twif, This Week in FTR, which is a weekly broadcast that highlights some of the research going on in our lab and in our building. And for our inaugural episode, what I thought I would do is talk about simple genome browsing, some stuff I've been doing with Integrated Genomics Viewer, and how to quickly render genomic tracks. And I'm going to use some of the QPX data I was working with over the past week to show you how to do it. To start from the beginning, if you don't have it installed, what I do is simply just download the source code to my hard drive. And what it does, it installs a zip file with about 7 files on them. This is what it looks like onto my hard drive. And then to start the Integrated Genome Viewer, I just double click on IGV.command, and what it's actually doing is opening up a terminal window, and then it's automatically opening up the window that you work in, the browser window. So the first thing I want to do is bring in a genome, and the way I would do that is at the top menu, which I guess you can't see here, but you would go genome, load genome from URL. And because that index is now there, it's loaded. So this is has about 500 sequences of the QPX genome. So now I want to add a different track. So we can look in here. Some of these are a simple bed file. I will go ahead and load that. So you can just copy the link address. And then in this particular case, I just go to File, Load from URL, paste in that bed file. And this is a bed file of SNPs. And this is what the bed file actually looks like. So it's got the scaffold name or the contact name, the location, and then what it is, what type of SNP it is, and then the proportion, and also the coverage. Really, all you need is 4 columns to be able to visualize that. So let's bring in another track. So we can also bring in GFFs. So this is a GFF. This is based on a blast in of a different species. These are actually, this is the E value, this is location, this is the ID. And so once again, we can just bring in that URL. And there we see that. And then finally, I just want to show a way to look at RNA-seq data, which is very interesting. So created an RNA-seq file in CLC, exported it as BAM out of CLC, and then just used the genome to coverage and bed tools to create a bed graph. And this is what the bed graph looks like. It's got the contact name, location, and then just the number of basically coverage. This would be very low coverage. I can do File, Load from URL, but this bed recognizes the bed graph. And this will take a second to load. It's a little bit bigger. And it also is a nice illustration of a different type of file. There you see that you can play with this a little bit more. Do a heat map. For these particular ones, I also liked to change the range too, so I don't really want to see anything below 20, so 28 below 28x coverage. I don't really think it's that necessarily that important. That's what it looked like. So you could pull this in, essentially RNA-seq data, and look at it in terms of where the genes are, what they're located, and compare them in a visual way. So this is just a quick kind of introduction to IGV and how you might use it to look at different types of genomic features. Tune in next week for another exciting edition of Twif, This Week in FTR.